Hello, my name is Jacob. This is Exam2 Essay2 for Bio461. I'm going to go ahead and help answer some of these questions within this unit. So the first part is to describe chemoreceptions of smell and taste. I'm going to start with taste, actually. So we have two different types, I guess more like three. We'll start with salty and sour tastes. So these are leaky ion gated channels, which includes sodium channels and hydrogen channels. Sodium is associated with the salty taste receptors, and hydrogen is associated with the sour ones. So when sodium is within the extracellular part of these cells or these receptors, it'll pass through a leaky ion channel, and when that enters into the receptor, that will cause a action potential or some sort of depolarization of the receptor, and this allows both leaky potassium channels to exit and unlock a voltage-gated calcium channel. Now, this voltage-gated calcium channel allows, obviously, calcium in, and that helps to bind to the snare proteins of neurotransmitters, which will be released to the actual synapse of a neuron, and that's how your brain will then determine that as a salty taste. And as for hydrogen, everything is actually the same, except it will close the leaky potassium channel, which will create a greater difference of depolarization in the cell. And again, voltage-gated calcium allows calcium in and releases those neurotransmitters. And the reason why sour tastes are, we can recognize them as different than salty is because of that greater depolarization, basically. And then we have the rest of them, which are sweet, umami, and bitter. So basically, those ones are ligand gated, or I guess ligand receptors. They don't use the ions into the cell, but in fact, a ligand, which will attach to a type of compound on the receptor of the cell, and this will release a alpha subunit. This alpha subunit goes and becomes this cascade using IP3, which will eventually use the IP3 to unlock a intracellular, or the sarcoplasmic reticulum, that contains calcium within the cell already, instead of using a voltage-gated calcium channel. And so this IP3 will then bind to a RYR receptor, or ryanodine receptor, and to release this calcium into the within the receptor, within the cell. Then, such as the other receptors, the calcium will then bind to snare proteins to release vesicles of neurotransmitters and synapse with the neuron, which would then interpret the brain as any of those things that I mentioned. So bitter, sweet, and umami flavors. So smell, how that works is we have a olfactory bulb that houses the body of the cells, body of neurons. These neurons then come down through the passing a bone plate and into the cavity of the nasal cavity, holding a receptor neurons on the ends of them, and there are tons of these neurons. And when those smells, or I guess those particles of smells, enter the nose, it will then attach to these receptors and then allow the neurons to recognize these receptors as very specific things. And so we have the ability to sense way more smells than we do taste. And even we're able to, I guess, recognize things that we haven't smelled in a long time into our memory. And another thing is that smell receptors are able to grow back or multiply, I guess, if they happen to be cut, at least in a non-permanent way. So yeah, that is for smell and taste. Now the next part of this is going to talk about describing how energy carried by sound waves turns into mechanical vibrations, to fluid waves, and then chemical signals to finally action potentials for the things we hear. So we have 3 parts of the ear per se, or the anatomy of what we hear, how we hear, I guess. You have your outer ear, which is the exterior of your ear, including the ear canal that leads to the tympanic membrane. Between the tympanic membrane and to the next part, we have these 3 basically mechanical parts, which allows the vibrations that enters our ear to amplify into the last part. So this middle part we just mentioned with 3 parts is the middle ear. And then the last inner part of the ear holds the actual receptors and lots of other actual parts that we'll mention as well. So these parts include, should include a type of membrane that will curl, and as the membrane extends into that curling piece, it actually becomes more thicker. It is filled with a lot of different fluids, and so thus meaning that those sound waves, in order for it to penetrate or be transferred into this fluid better, we have the middle ear parts that will amplify those vibrations. And so these fluids will then actually enter in, and or actually these will house the things that we'll need to hear, basically, which is our hearing apparatus. Okay, so the apparatus contains the cochlea. The cochlea is a structure that houses most of these other parts, such as the scala vestibuli, scala tympani, and the basilar membrane, as I was mentioning earlier about the membranes. And inside the cochlear duct, there are hair cells that basically are lining up just above the basilar membranes, and they reach up into the tectoral membrane, and how it interacts with this tectoral membrane when it comes to vibrations will then determine if neurotransmitters are released and synapse with neurons, and then will become those action potentials will become sound that we hear. So we have tons of these, and each are set to different sensitivities or different magnitudes of sounds, if you will. And these all run off of, mechanical potassium channels, which are at the top of these hair cells. And when these hair cells react, what basically happens is the basilar membrane will then push the hair cells up into the tectoral plates, and these finger-like structures on the top of the hair cells will then touch the tectoral plate and be moved and have a kinetic, some sort of energy. This kinetic energy will then open these mechanical potassium channels, and the potassium, following its gradient, will then create a depolarization of these receptors, as with other things, and allow in calcium to enter the cell or the receptor. This calcium will then attach to the snare proteins that's connected to the vesicles holding neurotransmitters, and the neurotransmitters would then be pulled in out of the cell and released towards the receptor of a neuron. And thus, like I mentioned, the neuron will then interpret it as a sound, and that's how we can hear things. Okay. The next part is we want to talk about how eye focuses light. So the anatomy of an eye is let's see. We have a circle essentially. The very outer part of the eye is the cornea, and inside of the cornea, kind of more towards the I guess the inner part of the eye, we have a lens, and this lens is a convex shape, meaning when light enters it, it'll bend it to a more acute point. And so when this light gets bent from the lens, it should hit the back of our eye at a single point. These two waves come together and meet. And that's how our eyes kind of is able to focus on the things we see. And if it doesn't hit at a single point in the back of the eye, such as it doesn't meet together or crosses too far, that's how we have blurry vision, and that's when people will need to be prescribed a prescription of glasses to be able to see. And basically what that does is have a convex or concave shape that allows to bend the light in whichever way it needs for that specific person to refocus that light to the eye. And that's how you're able to see more clearly, per se. The lens of the eye that bends that light is suspended by suspensory ligaments. It is not technically connected directly to the house of the eyeball, but it's suspended with these ligaments, which ligaments are connected to muscles that allows it to pull on the ligaments, the suspensory ligaments, and change the shape of the lens to either being more convex to a little bit less convex. And this is what we do typically when we have to see from closer to farther away objects. An example is reading or seeing something really far away. Then how we interpret this light to our brain exactly is, so when the light enters our eyes, it will hit photoreceptors that we have. We have cone receptors and we have rods. And if I remember correctly, cones allow us to visualize color, and rods are able to help us visualize light and dark things. Now these cones and rods are arranged in very specific manners, where different sizes, but basically in almost a bullseye shape, where we have a central photoreceptor and you have surrounding photoreceptors around it, like I mentioned, in a bullseye. And you have different sizes of these bullseyes because you have things you want to be able to focus on more acutely, and then things that can be, in your peripheral vision, just more, let's say, abstract. You don't need to be able to focus on the things you aren't looking directly at, right? So that's what happens there. And then same with these rods and cones, they allow you to visualize things in lighter / darker scenarios or places. And sometimes these photoreceptors, they're either activated through light or activated through dark, per se. So let's see. Yeah, and the amount of action potentials that occur from either those photoreceptors that react to either light or darkness, whichever they may be, they will have action potentials. And the amount of action potentials you get from those photoreceptors will help you to visualize exactly what you're seeing. And that's how your brain interprets sight, if you will. Let's see. yeah, so the mechanism of which is pretty long, but anyways, these photoreceptors are connected to horizontal receptors that goes towards, it all falls into the central photoreceptor. That photoreceptor in the middle will then release, or it may not release neurotransmitters towards the bipolar cell. And the bipolar cell will then interact with these neurotransmitters in whatever way it may be to then release glutamate is the neuroreceptor we use here, the neurotransmitter, sorry. The neurotransmitter of glutamate would then be released towards a ganglion cell. This ganglion cell is the neuron that basically creates these action potentials with what we see and sends it to the brain. Okay. We want to talk about hearing as well, the mechanisms of how we hear. I'm sorry, we probably already did this. Yes, so like I mentioned earlier, we have the hair cells within the cochlea of the ear, which is the innermost part of the ear, like I mentioned. And the basilar membrane will then push these hair cells into the trochlea, and it is activating mechanical potassium receptors or channels by these hair-like protrusions from the cell to be moving and pulling on the mechanical channels to open it, basically. Potassium will go down its gradient. It has a big driving force of 120 millivolts. And so allowing these potassium to move down its gradient creates this depolarization, causing calcium to enter, and calcium pulling on the snare proteins to release these neurotransmitters from the vesicles. And these will then be released towards the cochlear nerve and be received by the brain as action potentials to interpret it as a sound, basically. All right, that is my Essay2 Exam2 question there. Thank you for listening.